All right, guys, welcome back to Underground Science. And in this video, we'll be talking about toll-like receptors 7 and 8. All right, so let's go and get started. So, toll-like receptors 7 and 8 usually responds to single-stranded RNA, right? Viral single-stranded RNA. Okay, so let's go and see how these toll-like receptors 7 and 8 get activated and then what that leads to. All right, so let's say, well, first we should know that toll-like receptor 7 and 8, right? Where can you find these receptors? You can find these receptors in B cells. You can find these receptors in, let's write that better, in B cells. You can find these receptors in dendritic cells. And of course, you can find these receptors in macrophages also. Okay, so these are some cells that you find toll-like receptors 7 and 8 in. Okay, so and you can find these in macrophages. So remember that when we talked about toll-like receptors 1, 2, and 5 and 6, right? We talked about how you can find those toll-like receptors on the cell surface, okay, even toll-like receptor 4. But remember when we started talking about toll-like receptor 3, we switched to the fact that you don't find that on the cell surface anymore. Now you start finding that on the inside of endosomes. Okay, so same thing goes to toll-like receptors 7 and 8. You actually find toll-like receptor 7 and 8 inside endosomes, not on the cell surface. Okay, like you would, for example, for toll-like receptors 1 or 2 or 4 and so on. Okay, so let's go and see how these toll-like receptors can get activated. So let's say you have a virus with single-stranded RNA, all right, and you have a macrophage here or a dendritic cell, right, that sees this virus. Of course, it's going to try to phagocytose this virus and take it in, right? And then it's going to take it in. So draw that, draw this macrophage a little bigger. And then you're going to have an endosome here. And then, right, so we know you form your phagosome. And you're going to have the virus right here with its single-stranded RNA. Okay, so once we phagocytose this virus, right? Inside the endosome, remember we said we find what toll-like receptors? 7 and 8. Okay, so let's say this is toll-like receptor 7 and this is toll-like receptor 8. Okay, so what's going to happen is this virus, right, this single-stranded RNA will actually will get exposed to these toll-like receptors on the inside of the endosome. But we have to remember, right, for the virus or for the single-stranded RNA to get exposed, we have to break down this covering, the capsid, right? So what actually happens is the pH inside the endosome decreases, okay? The pH inside the endosome decreases, and this will actually help break down the capsid of the virus so that we can actually expose the single-stranded RNA to the toll-like receptors, okay? So once the pH decreases, we can activate certain proteases or cathepsins, right? These are just proteins that will help degrade the virus, the capsid of the virus or the envelope of the virus, okay? So once that happens, now, so let's erase this covering, right? So to show that, okay, we have now degraded the covering of the virus, the capsid or the envelope, right? And then so then this will show us that, okay, now the single-stranded RNA is exposed to the toll-like receptors. So once that happens, this single-stranded RNA molecule, right, can get the toll-like receptors can recognize the single-stranded RNA and get activated. So let's say we said that this was toll-like receptors 7 and 8, right? So if the toll-like receptor binds, let's say to 7, or not, so, excuse me, if the single-stranded RNA binds to toll-like receptor 7, then that's going to lead to the dimerization of 7 and 8. Okay, so let's go ahead and write that down. So let's say we show that this single-stranded RNA, right, will bind to toll-like receptor 7 or 8, okay? Once this happens, we're going to get dimerization of both 7 and 8, which we can draw something like this, right? Showing that both 7 and 8 come together. All right, so let's just go ahead and like color this in so we can show that, okay, both of them came together, right? It's probably a horrible representation or horrible drawing of dimerization, but I'm not good at drawing. So this is going to be our dimerization of 7 and 8, okay? Of toll-like receptors 7 and 8. Once this happens, this we have activation of these toll-like receptors now, okay? Now, once these toll-like receptors get activated, we get the activation of the MyD88 signaling pathway. And we already know from our previous videos, right, especially our cell surface toll-like receptors, that MyD88 leads to the activation of nuclear factor kappa B, which then leads to the release of many different cytokines, right? Because remember, nuclear factor kappa B goes to the nucleus and leads and increases gene expression so that you make a lot of different cytokines, right? So you make different cytokines like tumor TNF alpha, right? You make cytokines like interleukin 1, interleukin 6, interleukin 8, interleukin 12, right? And so on. So this leads to a bunch of cytokine release, and we know that cytokines contribute to the overall process of inflammation, and they attract different white blood cells to that area of inflammation or infection, right? But what also happens when toll-like receptor 7 and 8 get activated, yes, they do activate the MyD88 pathway, but they also activate the IRF7 pathway, okay? Or the IRF7 molecule. So MyD88 activation, okay, leads to the production of cytokines. All right, so it leads to this pathway right here. But what also happens is when MyD88 gets activated due to toll-like receptor 7 and 8 activation, then this also leads to the activation of a molecule called IRF7, okay? And then IRF7 leads to the production of interferons, right? Or type 1 interferons. So it's going to lead to the production of interferon alpha and interferon beta. And we know from previous videos, right, that interferons are good for inducing an antiviral state. So it helps reduce the viral spread to nearby cells. Okay, so it's going to help induce an antiviral state, whether that be from producing antiviral molecules like protein kinase R, right, which we talked about in our previous videos, helps reduce the translation of certain viruses, or interferons can help increase MHC1 expression, right? And we know MHC1 expresses viral particles, right, onto its cell surface, which then helps CD8 cells destroy that cell so you can contain the virus. All right, so different tasks, different tasks like that help reduce viral spread, and that's the job of interferons. Okay, that's why you can give interferons for certain viruses, for certain viral infections like COVID or hepatitis or something like that, so you can reduce the viral spread. All right, so that's the importance of the IRF activation. And then we have cytokine release and production also, which will help attract white blood cells and increase the overall process of inflammation and vasodilation and swelling and all that that we know, right? Which is the normal um, steps of inflammation. Now, if we were talking about something like influenza virus, right? And you took let's say we were talking about our alveoli, for example, right? Or our airway in general. Let's say there's the influenza virus, right? And we have our macrophages, right, in our alveoli. So we can take up that influenza virus and it would lead to the same events because what happens is the influenza virus is single-stranded RNA in it, right? Viral single-stranded RNA molecule inside of it. So if we drew that, let's say this was our influenza virus and it has our single-stranded RNA, right? If the macrophages take this in, it's going to lead to the same process. So this is a like a clinical example, right? A real life example of how of how toll-like receptor 7 and 8 work. Okay, so if it takes in the influenza virus, right, or phagocytoses it, then of course we're going to get our endosome formation or our phagosome formation, right? So it's this our macrophage, and then we endocytosed it. So now we have our phagosome or endosome, right? And then we have our influenza virus in it with its single-stranded RNA molecule. So the same thing will happen is the endosome, right, will decrease its pH inside of it, which can help activate certain enzymes like proteases to degrade the covering of the virus, right? If that happens, now the single-stranded RNA molecule, the viral single-stranded RNA molecule, can get exposed to the toll-like receptors on the inside of your endosomes, like what? Like toll-like receptors 7 and 8, right? So if that happens, then we already went through this, right, in the last slide, that will lead to the dimerization and the release of many cytokines through the MyD88 pathway due to the activation of nuclear factor kappa B. And we said IRF7 will get activated, leading to the release of interferons like interferon alpha and beta, which will do what? It will reduce the viral spread. So it's going to induce an antiviral state. All right. So that's just a nice clinical example of how toll-like receptor 7 and 8 get activated and what's the use of that. That's why toll-like receptors are important, right? Because it's going to help our immune system fight off whatever virus or pathogen is infecting us. For example, SARS-CoV-2, right, has single-stranded RNA in it, and it can bind onto toll-like receptor 7. And we know when that happens, we went through the result, right? Activation of nuclear factor kappa B, release of many cytokines, and release of many interferons. Now, people who have toll-like receptor 7 mutations, right? If you had a toll-like receptor 7 mutation, then that means toll-like receptor 7 wouldn't work properly, which means you wouldn't be able to secrete proper amounts of interferon, right? And if you can't secrete proper amounts of interferon, then you can't have a proper antiviral state. Okay, you can't reduce the spread of the SARS-CoV-2 virus proper as well as other people can who don't have toll-like receptor 7 mutations. Okay, so you can't have a proper antiviral state, which means the SARS-CoV-2 virus can infect these people at a greater extent than people who don't have toll-like receptor 7 mutations. So it's important to know how toll-like receptors work because you can apply a lot of, or you can there can be a lot of clinical applications to these receptors too. Okay, just as we saw with certain viruses like influenza and SARS-CoV-2. All right. So I hope this video helped a little bit and made the pathway for toll-like receptors 7 and 8 more clear. Thank you guys for watching, and I hope to see you guys in my future videos.